Hi everyone, it's Dr. LeBlanc. The big picture for translation is that protein production systems or expression systems are very widely used in the life sciences, biotechnology, and medicine. For example, genes that encode a particular protein can be isolated from host organisms and then inserted into a variety of protein expression platforms such as E. coli, insect cells, mammalian cells, and yeast cells. Molecular biology research uses many proteins and enzymes which are from expression systems. For example, the DNA polymerase for PCR, reverse transcriptase for RNA analysis, and restriction endonucleases for cloning are all produced by host organisms that encode a recombinant gene. Here's an example. The Taq polymerase that we used in lab for our own purposes was made from a recombinant source. In other words, E. coli produced the DNA polymerase that was originally encoded by Thermus aquaticus. Protein expression systems also exist for drug discovery to make proteins that are screened as biological targets or as potential drugs themselves. Here's a variety of different protein expression systems that exist in the commercial venue, from Thermo, from Sigma Aldrich, from New England Biolabs. There are many more biotech companies that produce protein expression systems. And here's an article that talks about bacterial expression systems, E. coli, and beyond. At the European Molecular Biology Laboratory, it's a big institute similar to NIH that exists in Heidelberg. There's a core facility that will clone the gene for the protein that you're interested in and purify it for you. They focus on E. coli expression systems, but like we just saw that there are many other host organisms that you can express proteins in. There are also very significant applications for expression systems in industrial fermentation, biopharmaceuticals, and for such molecules as insulin to treat diabetes and other enzymes and proteins like TPA, tissue plasminogen activator, which dissolves blood clots in heart attack victims. So how do you make insulin? How do you make human insulin in E. coli? Why do you make human insulin in E. coli? Well, in the early 1920s, it was discovered that if you extracted insulin from cows and pigs, from their pancreas, it would control diabetes in humans. However, humans are not cows and pigs, and so allergic reactions often happen. So the solution was to clone the gene, isolate the human gene, and insert it in E. coli and trick E. coli into producing human insulin. This is the diagram that shows how this has happened. You take DNA from a human source and you take a plasmid from a bacterial source and recombine them in the test tube to make a recombinant DNA molecule. So the red part is bacteria, and the red part is human, and the blue part is bacteria. You put this back into a bacteria cell. The bacteria will then transcribe the gene into messenger RNA and then translate the messenger RNA into protein, insulin. You put this in a giant fermentation tank to make lots and lots of it, and then you extract it and purify it and bottle it up for human consumption. Instead of growing it in flasks in the lab, you can grow it in these giant fermentation tanks, and many biopharmaceutical companies have facilities with many of these to produce large quantities of human insulin for the human population.